Hi everyone, a very good morning to everyone. I'm Sritaran Vasu from DBT4B. Okay, the reason I'm here is to do. Okay. Hi everyone, today I'm here to doing a simulations about bacteria enumerations. Okay, to carry out the. Hi everyone, today I'm here to carry out the bacteria enumeration simulation. in order to uh in order to okay Hi everyone, here I'm Sritaran Vasu from DBT4B, here to doing a simulations about bacteria enumerations. First of all, bacteria enumerations is involve the counting of bacteria cells. So we using a variable cell count plate technique here. So first of all, click on the blackboard to view a large board discussion. So enumerations of bacteria is a part of daily routines as laboratory microbiologists often has to determine the number of bacteria in a given sample, as well having to compare the amount of bacterial growth under the various conditions. Enumerations of microorganisms is of special importance in dairy microbiology and food microbiology and water microbiology. Knowing the bacteria count in the drinking water, fresh milk, buttermilk, yogurt can be useful in many aspects of industrial microbiology. As you guys know, bacteria are so small and numerous, count them directly can be very difficult, so diluting of samples to point at which number of bacteria has been reduced to very small number, which is we using a serial dilution method, which can reduce the concentrations of microbes in the sample. That enables us to visualize the small amount of microbes on the plate. So next, so there is three links. Variable plate counts is a count of viable or live cells. It's based on the principle that um, viable cell replicates and give rise to the visible. We can summarize the via plate count is a method where measure amount of liquid cultures inoculate on the plate, and the plate incubate, and the colonies that result by visualize on visualize it by naked eyes. The reasons why variable plate count criteria is to identify the number of actively growing or dividing cells in sample. Okay, so we move to next. The reasons why the plates having 30 to 300 colonies is chosen because the range considered statistically significant. If there are less than 30 colonies on the plate, small errors in dilution techniques or presence of few contaminants will have drastically effect on the final count. So in case there are more than 300 colonies on the plates, there will be pure isolations, and colonies will have grown together. So generally one want to determine the number of colonies per milliliter of sample. So to find this, the number of colonies is multiplied by the number of time the original ml of bacteria was diluted. This is procedure that we are going to use in variable plate counts. So will be there are four samples that we are going to test in this simulations, which is water from drinking fountains, boiled water from drinking fountains, water from local river, boiled water from local river. Okay, per meal is the link. Okay, so okay, first steps, take a 6 dilation tubes, so where each tubes containing 9 ml of sterilized saline. So we click on the dilation tube, right, to bring them into the table. So why the saline solutions is used, why not water? The reason is because The water is a hypotonic in nature, so the concentrations of solutes greater inside the cell than outside of the cell. So this water will try to enter the body of bacteria cell, which can damage the bacteria cell. So while saline is a high isotonics in nature, will remain the concentrations of solutes both were inside and outside of cell, will also remain the osmotic pressure in the bacteria cell. So next, click on eyedropper. We draw 1 ml of sample. There is 4 sample. As I told you before, water from the drinking fountains, boil water from drinking fountain, water from local river, boiled water from the local river. So before this, click the water sample to bring the sample. Now click eyedropper to withdraw 1 ml of sample and dispense this to the first tube. So click next when the initial transfer is finished. So click again click again on the eyedropper to withdraw 1 ml from first dilutions tube. You can use whether the same dropper or you can change this eyedropper. Okay, continue doing this from tube to tube until the dilution is complete. Complete through dilation tubes. So click on next when the dilation is complete. So dilutions of each of dilutions can summarize in the image. So dilation factors of tubes is listed. So here is the dilation factors for each tube. So click on next when you're ready to start. So next we will be inoculating agar plate with the last 3 probe culture dilutions where 10 power of 10 and 10 power of 4 the last 3 tubes, the last 3 culture dilutions tube were 4, 5 and 6. So click on the agar plate on the shelf to bring them to the table. Now click on the eyedropper to transfer 1 ml of dilutions plate, 1 ml of dilutions plate number 5. Next, click pencil. So after you label plate 1 with dilutions 10 power 4 and plate 2 with 10 power 5 and plate 3 10 power 6. Once you're done, click the pencil to label the agar. Make sure label correctly the dilution factors and the plate number. Once done, click next. We are still on the sample 1 where it's fountain water. Okay, click on the agar plate to place them in incubator at 37th degree for 48 hours or 2 days. We will now need to perform the same dilutions and inoculation step for each step samples. The process is same for each samples, and then assume the process of dilutions and inoculations has been complete for all of water samples, and then 48 incubations time has now been completed. So incubate or at 37 degrees, which is room temperature for 2 days. then click next when you're ready to view incubated plates. So click on the incubator to bring all the agar plates to. Okay, these agar plates all are incubated. So each of group of incubated plates is labeled with source of their respective sample. So now we're going to click one of the sample where we click on the first sample is where found which is fountain water. Click here. So if you want to, okay, we're going to view all the agar plates. So this is what we get. So the next plate where is 10 power of 5 dilation plate. So next sample 2 boiled fountain water. So next is river water. So we're going to view the river water sample. The fourth sample is boiled river water. Okay, this is a sample for 10 power of 4.